Good morning, everyone. My name is Tabitha Shah, and today I will present on the regulation of aldolase expression, focusing on its role in glycolysis and glucogenesis, the different isoforms, and how aldolase B expression is controlled by specific transcription factors in various tissues. Let's start off with glycolysis. Glycolysis is a fundamental metabolic pathway that breaks down glucose into pyruvate, releasing energy in the form of ATP. ATP, also known as adenosine triphosphate, is the primary energy currency of cells, critical for powering many biological processes. This pathway occurs in the cytoplasm of nearly all cells and is essential for cellular energy production, especially under anaerobic conditions. Glycolysis involves a series of enzyme catalase reactions to convert glucose into two molecules of pyruvate. Aldolase plays a key role in glycolysis by splitting fructose, 1,6-biphosphate, into two 3-carbon molecules, glycerol 3 and phosphate and diacetone phosphate. This step is essential because it breaks down glucose into smaller parts that can be further processed to release energy. To start glycolysis, the cells invest ATP to phosphorylate glucose, which helps trap it inside the cell and prepares it for breakdown. Later, the pathway generates ATP that fuels cellular function. Aldolase A is especially important in high-energy tissue, like the muscle and brain, where rapid ATP production supports intense activity. This illustrates how aldolase isoforms match tissue-specific energy needs. Now, in contrast, gluconeogenesis is a process of synthesizing glucose from non-carbohydrate sources, such as lactate, glycerol, and amino acids. This pathway mainly occurs in the liver and kidneys and is critical during fast or intense exercise to maintain blood glucose levels. It essentially reverses glycolysis, but includes some unique enzymes to bypass irreversible glycolytic steps. Glycolysis and glycogenesis are two important processes that control the body's energy and blood sugar levels. Both use an enzyme called aldolase, which either breaks apart or joins specific sugar molecules. These processes happen partly inside of the cell and share some of the same enzymes. ATP, the cell's energy molecule, is important in both glycolysis, which uses a little ATP at the start, but then makes more, while glycogenesis uses ATP to build new glucose. Even though they use some of the same steps, their jobs are opposite. Glycolysis breaks down glucose into smaller parts to release energy, especially in muscle and in the brain, where aldolase A is common. On the other hand, glycogenesis makes glucose from sources like lactate, mostly in the liver and kidneys, to keep blood sugar steady when we haven't eaten. This process needs energy instead of making it. The different types of aldolase matches these roles. Aldolase A works in tissues that need lots of energy from glycolysis, and aldolase B works in tissues that make glucose through glycogenesis. Aldolase B is carefully controlled by specific proteins called transcription factors that help the body adjust glucose production based on its need, and even the day and night cycle. Together, glycolysis and glycogenesis balance each other to keep energy and blood sugar steady as condition changes. Now, let's look at aldolase. This enzyme helps break down a 6-carbon sugar called fructose, 6-phosphate, into 3-carbon sugars. This step is very important because it splits sugar into parts that cells can use to make ATP, the energy molecule. Aldolase works during glycolysis, especially in tissues like the muscle and brain, where energy is needed at high levels. The aldolase enzyme helps the body get energy from glucose by breaking it down step by step. There are 3 isoforms of aldolase, A, B, and C. These isoforms differ in amino acid sequences and tissue distribution. Aldolase A is predominantly found in muscles and in brain. Adelase B is mainly found in the liver, kidney, and small intestines. And Adelose C is primarily expressed in the brain. Adelase A supports high energy demand tissues like the skeletal muscle and red blood cells, where rapid glycolysis is needed. Adelase B plays a critical role in liver metabolism, especially fructose metabolism and glycogenesis. Adelose C is involved in brain-specific metabolic processes, although its exact function are less well understood. Today we're going to focus on Adelase B. Adelase B is very important in liver, kidney, and small intestines because it helps with two main jobs: making glucose when the body needs it, glycogenesis, and breaking down fructose from food. When we eat fructose, a sugar found in fruit and many sweeteners, the liver changes it into a molecule called fructose 1-phosphate. Adelase B then splits this molecule into smaller parts. These smaller parts can now enter the pathway that either produces energy, glycolysis, or makes new glucose, glycogenesis, depending on what the body needs. This process lets the liver use fructose effectively, either for quick energy or to keep blood sugar steady when we haven't eaten for a while. The body controls how much Adelase B is made by special proteins called transcription factors. These factors act like switches, turning the Adelase B gene on or off based on signals like whether we are eating or fasting, and even the time of day. For example, HNF1 and HNF3 help start Adelase B production by attaching to its gene. CEBP adjusts depending on food availability, and DPA links Adelase B levels to our body's biological clock. Since glycogenesis uses energy, it's important that Adelase B is carefully controlled to keep the body's glucose and energy balance. Adelase B's special role and careful regulation let the liver, kidneys handle sugar in a way that matches the body's needs, helping keeping blood sugar steady and energy always available. The body controls how much Adelase B is made by special proteins: HNF1, HNF3, and CEB and DBP. These factors are switches. FNH1 and FNH3 work by acting directly to Adelase B's gene control areas to turn on its production. This ensures that Adelase B is made when the liver and kidney need it most. CEBP helps adjust Adelase levels depending on whether the body is fed or if it's fasting. For example, during fasting, when the body relies on making glucose, CEP increases Adelase B production to support glycogenesis. DBP connects Adelase B levels to the body's internal biological clock, or the circadian rhythm. This means Adelase B amounts go up and down at different times of the day to match the body's changing energy needs. Because glycogenesis uses energy, it's important that Adelase B is carefully controlled. This control helps the body balance when and how much glucose is made, so energy is available all times of the day and is not wasted. Together, these transcription factors cooperate to precisely fine-tune Adelase B expression, ensuring that sugar metabolism in the liver and kidney adapts smoothly to changing physiological conditions, such as feeding, fasting, and daily energy needs. This careful regulation helps maintain stable blood glucose levels and efficiency energy use. Thank you so much, and all my references have been listed. Have a great day.